Hello, everyone. I see people are still filing in, but we're going to go ahead and get started because there's a lot of great content in this talk for everybody to learn about and to ask questions about. So the format for today is I'll give an introduction, introduce our panelists, who will also give some very good information for all of you. And then we'll open it up to questions to the panel, both questions that I have and questions that you might have. So I encourage everybody to come to the microphone, introduce yourself, and let us know what's on your mind. So with that, thank you for coming to this session. I know we've got some stiff competition next door. So if you're here because you really want to be, so we'll make it happen for you. So we're talking about cord blood as a platform technology supporting emerging cell and gene therapies. Platform technologies really is kind of a term that's been used in the computer world, in the software business, but it connects developers with resources and end users and creates a system that leverages the strengths of all the partners. So if you think about it, you have the developers, you have the consumers in the middle, and then you have the other folks in the middle who are then leveraging all sides. So anyway, it provides speed, agility, and ultimately new therapies to treat patients in our setting, in the cell therapy setting. And we know that alternative uses, we've heard a lot about them at this conference in terms of creating cord blood bank stability, sustainability. And so I think this talk is really going to follow along with that. So the objectives here are, how do cord blood banks diversify and expand their cord blood inventories to prepare for participation in this new realm? We know that cord blood banks have a little bit different business model than the commercial partners that we're going to be working with. And so it's important to be able to prepare for that. So I have my computer up here because we have some very distinguished partners here in our panelists, and I wouldn't remember or do them justice on their introductions without it. So first we have Christina Malif. She has a PhD from Stony Brook University in genetics and is a classical developmental biologist and glycobiologist. Before joining Bloodworks to form their new department, Bloodworks Bio, she worked as a technical consultant for Miltigny, where she developed a passion for human stem cells and immunology, working with clients in the Pacific Northwest. She believes the future of cell and gene therapy outside of rare disease indications depends on the resilience and the growth of cord blood banking industry, and is excited to be a part of it. Next, we have Wouter van 't Hoft, and I put a T there that wasn't necessary. He holds a PhD in cell biology and has over 20 years of biotech experience in translational development of adult stem cell therapies and bone marrow, stromal cells, and HPC cord blood. As the director of the cord blood bank at the Cleveland Cord Blood Center, he helped to obtain a BLA approval from the FDA for HPC cord blood, which is called CleveCord. That was in September of 2016. And since 2020, he leads the Cleveland Cell Therapy Incubator, which is a subsidiary of the Cleveland Cord Blood Center, to develop programs for diversified use of cord blood in cell and gene therapy beyond hematopoietic transplant. And then we have Dr. Heather Stefanski. Dr. Stefanski joined the NMDP in May of 2021. I couldn't have been more delighted to have her come on board with us. Prior to her arrival, she was an associate professor of pediatrics at the University of Minnesota and was actively involved in clinical care and research involving children with life-threatening blood and immune system disorders. One of her areas of focus is the use of cord blood units in stem cell transplants and other cell therapies. She's board certified in both pediatrics and pediatric hem-onc, and her title right now is Vice President of Medical Services at NMDP Be the Match. So with that, I'm going to leave here and let them take over. So Christina.
So hi, I'm Christina Malif. Thanks for having me today. My background is a little bit different than most of the people here. So I am business development for Bloodworks Bio. So I not only cover cord blood, but I cover mobilized apheresis, whole blood, and isolated cells. So I'm coming in from that perspective of making a sustainable business. So a huge part of expanding into providing support for the cell and gene therapy space is really understanding how the space and providing cords for it fits in with your mission and vision. Bloodworks Bio is part of the Bloodworks Northwest, which was formerly the Puget Sound Blood Center, and so I thought I'd explain how we evolved into cord blood in the first place, and then show you exactly how we're evolving into supporting cell and gene therapy. So Bloodworks Northwest has been around for over 75 years and started as a blood bank, and our mission is to provide a safe and reliable blood supply for the Pacific Northwest. However, over the last 75 years, we've identified that the definition of blood and the way it can be used as a public health partner has changed. And so we've changed how we operate and what we provide over time to meet the rising needs in the industry. So that has involved opening a public cord blood bank, which we will talk about today, as well as research donor programs to provide starting materials for both research and clinical manufacturing in the blood-derived cell space. So today I'd like to show you exactly what we did to evolve into this space as a cord blood bank. So today I'm going to talk to you about understanding the language and the different platform technologies that are available, understanding your inventory and what you're capable of providing, how to operate with sound financial practices, which I find is a very big issue in the public cord blood banking space, picking your niche for growth and how you provide support to corporate clients as well as universities and biotechs, and then also understanding your abilities and what you can and cannot do and how you should address those things with clients. So let's start by talking about platform technology. When somebody approaches you for cord blood, they have a lot of options, not just cord blood. So typically when I'm having a sales conversation regarding cord blood, I'm also talking about mobilized apheresis, whole blood, and bone marrow, because anybody coming to me for cord blood has the opportunity to use any of these other starting materials. I personally believe that cord blood is the only great option out of any of these for many reasons, including availability, ability to know your inventory in advance, and independence from having to recall donors who may or may not be available. But when a client comes to you, it's important that you understand the differences between these products and why cord blood is a valuable asset for their supply and production pipeline. And we can serve basically every industry within biotech and academia. So of these 6, I picked these 6 because all of them use cord blood, but all of them use different types of cord blood in their industry. So drug manufacturing and cell and gene therapy will have a need for cryopreserved clinical grade cords, whereas research and academia will need your throwaways that can't be banked. And diagnostics and drug development will oftentimes use your older cords or cords of exceptionally low volume. So by targeting clients in these 6 industries, you can find a home for every single cord that comes in your door, which has always been a personal goal of mine. So it's really important that you understand your inventory when talking to clients. It's very easy to say, well, we're a cord blood bank with 20,000 or 40,000 or 50,000 cords. But what does that actually mean? So I've put together a pathway to look at your inventory to understand what you actually have to offer. So the only non-negotiable on this slide is understanding your consent. Everybody in this room has had their consent go through an evolution over time. You'll have cords that are just for banking. You'll have cords that may be consented for commercial use, and you may have some that are consented for commercial use and may be used for iPSCs. But it's very important to understand and segregate your inventory based on what you can and cannot use them for under your consent. That's the top priority for discussion with any client. A second thing that you impacts most people in this room is their HRSA status. So if you are offering these for cell and gene therapy, do you need to replace them? I believe that this is a very personal question for each cord blood bank as to whether they're willing to replace units and offer these to clients. But for the sake of this pathway, I'm going to say that we're going to use non-HRSA funded clients, or cords. And then I would go through an internal evaluation. Does providing this cord to a cell and gene therapy partner align with our original mission of providing cord blood for transplant? So if this is a minority or high transplant demand, maybe high TNC, high CD34, a more likely to get used HLA type, then I won't make it available to cell and gene therapy customers. However, if it's low demand by those criteria, then I can start talking with clients about what their actual needs are for their starting material. And typically questions come in such as how were they processed? Was it a manual wash or did you use a CPAX? Do you have segments available and how many? And are you willing to sacrifice those is a common question. What additional samples exist and what can we get from those samples? And then what additional data do you already have available that I don't need to pay you to generate? Do you have genotyping? Do you have immunophenotyping? So really, once you start to drill down from your 20,000 starting cords, you're going to find that there are actually very few that are consented and you're willing to give up. So in order to sustain yourself independently of cord blood banking for transplant, it's very important to employ sound financial practices. And I was really surprised when I started talking with cord blood users in research and also people in the industry that they weren't employing the same financial practices that are common for people offering apheresis or mobilized apheresis, which was pretty shocking. So I'm going to tell you how I price my cord blood and the things that go into that consideration and how I make it sustainable for our cord blood program to expand our offerings. So the first thing I do, and I do this on a monthly basis, is I calculate my cost per cord. When I look at the cost of reagents, transportation, what I've spent on recruitment, collections FTE, the cost of accessioning the kit and deciding whether it's available for research or whether it goes into a banking pathway. And then I divide that over the total number of cords we've collected in the last 12 months. You can imagine that this varies highly based on things like gas prices and birth rates and your collection model. Are you paying people to collect or using volunteers? And so before pricing things out, I look at the variability over the last 3 to 5 years. So at a minimum, for every cord, even if it's just going for research, I have to recover my costs. So I do not price things lower than what it actually costs me to get that cord in the door and make a judgment of how to use it. Then when assigning a margin on that, I don't look at what the person next to me is doing. I go through and make a strategic plan of what I hope to accomplish over the next 2, 3, 4 years as a strategic investment. And then I back calculate and assign a margin and a price based on the frequency of the cord type, so how many are going to research, how many are really high quality that could be used for process development, how many could be banked, and then assign margins and pricing based on my financial goals to reinvest into the program. It's really important that when you're setting pricing for your cords, you're not trying to justify somebody else's pricing. And I see this a lot in platform technologies, and it's very destructive to the industry because our margins are our margins, our costs are our costs. And we can't predict how much variability there will be in recruitment or the birth rate or the success of collections. And so I don't look at what other people's costs are, I don't try to justify them. And if a customer asks, well, why are you charging us for a research cord that would go in the trash anyway? I say, well, it cost me to collect that cord and I value it. And it's something that if you continue on and use clinical cords, you'll have to understand the value of it too. And after that conversation, I rarely get any pushback. Something else to consider is the volatility in collection capabilities right now, given the turnover in staffing, the decline in the birth rate, shortages of CPACs, kits, and those types of things. When you look at other things that you purchase on a regular basis, whether it's gas or eggs or milk or airline tickets, those groups never apologize for the fluctuations in their pricing, just as you may need to adjust your pricing over time on a quarterly or annual or biannual basis. So when you're setting up pricing and contracts with your clients, it's important to take into consideration things that you cannot predict: supply chain issues, availability, staffing turnover, and be sure to structure your agreements in a way that you can adjust your costs if there are certain things that happen. Say your transportation costs go up 50%. If I can show that to a client, I have the right contractually to renegotiate that price, even if it's within a term limit. So a big thing in supporting this space is picking a niche to grow into. We've seen some really beautiful presentations by people who are isolating CD34s and expanding them, or going after different cell types. And so I want to talk a little bit about the types of niches that you could develop into and picking the right one for you. So most importantly is keeping in line with your bank's mission and vision, and also your ethics and your risk mitigation tactics. So it's very easy, presumably, to go into something like genotyping, but genotyping is expensive. So this would be something that you are pricing against in your other chords, or assigning a fee when you actually sell these chords. So you could go into genotyping. You could also go into isolated cells. That's something that we do and we're very good at. So we can isolate CD34s or CD56 or CD14 from these cells and preserve them as a research or a clinical grade product to corporate clients. You can also do cell culture. Cell culture is very different. It requires a certain amount of expertise and space, and has very specific requirements for your rooms and personnel. So that's something that's difficult to go into, but it's possible. And then you have to ask whether you're willing to progress past minimally manipulated cells into a modified gene space. So are you interested in doing small molecule derived iPSCs or CRISPR derived modifications of your cells? Furthermore, a separate question should be whether you're willing to accept the risk of introducing lentivirus or adenovirus or some type of viral transduction system into your facility. And if you have a separate facility, that might be a good option for you. Another opportunity would be to become a unit provider to other banks that are expanding to support these industries if you're not capable of growing at the time. So if you see a bank that needs 20 fresh cords to be pooled on a day and may not be capable of it, but they have the capability to do other things, approach them and offer to sell them your cords because they want to expand as much as you do. Another opportunity would be to provide more cords for research or provide cryopreserved cords for research. And this is something that we really don't address much. One of the things that I used to hear most frequently when I first started was, well, I get my cords for free, which is great. But every time I interact with a researcher, one, I need to recover my costs, but two, it's an opportunity to educate that person on what cord blood can achieve and the things that it's being used for. So I view those opportunities as building cord blood champions and recovering my costs. And one of the things that we typically hear as well, there's going to be too much competition, or I'm going to have to fight with this bank to win this contract and that type of thing. So I just very quickly mapped out capabilities of several cord blood banks that I know and redundancies in those offerings. So you can start to see very quickly that even with very large investment in development for cell and gene therapy space products, we're not expanding in the same ways, and there's very little redundancy. So if we have corporate partners who want, say, isolated cells with CCR5 genotyping with cell culture, there's only one person who can provide that. And when you start to look at the inventory available or look at the cost of prospectively banking those units, it becomes very expensive. So I would say it's probably going to take a decade with all of the cord blood banks growing in parallel to actually saturate these capabilities. And it's something to consider. If any one of our corporate clients hits commercialization, no one bank will be able to support their needs. And the last thing I want to discuss is actually understanding your capabilities. So sales conversations when you're dealing with these clients is very different than operating in a transplant space, because the people in transplant know what they can and can't do and what you are providing and what the standard is. That's not the case with clients looking to do cell and gene therapy. The most important thing is, if you are a licensed bank, understanding your licensure and knowing what you can and cannot sell your licensed units for. That's something that's commonly confused. And then I thought I'd walk you through a couple of terms that I hear most frequently when interacting with clients. So the first thing that I often hear is I'll have a customer come to me and say, well, I need a GMP cord. Well, fun fact, GMP cord doesn't actually mean anything. So if you look at the use of the term GMP for all of the other products that go into these cell and gene therapies, it's a branding term. It actually has no meaning when you're talking about a clinical reagent. Clinical also doesn't have a meaning. We'll talk about that in a second. So you get people standing in front of you asking for something that's GMP, and they don't know what they're talking about, but if they have expectations, they're not going to be met by your expectations. The second thing, and we have the luxury of using this term commonly with transplant, is clinical cords. So oftentimes you'll have somebody come in and say, well, I need a clinical cord. Great, are you doing a transplant? If the answer is no, that doesn't mean anything to me either. And oftentimes they'll have very different expectations based on their indication. But then you get to something like 21 CFR 1271, and it's easy to get very excited because you can say, okay, well, I'm subpart C compliant. So there's an actual regulation with an actual definition. And so that should tell you exactly what you can and cannot do, right? No. So it's unclear with current cord blood banking practices and the feedback being given to these clients, whether being 21 CFR 1271 subpart C compliant actually meets their indications for using it as a starting material for cell and gene therapy. In fact, what you'll find is that a lot of these clients want you to do upfront testing on the cord unit itself rather than maternal testing, because they don't want to do downstream infectious testing if they're doing culture and further manufacturing. So it's very important when you're in front of these clients to actually get specifications. The last thing that you hear is, don't worry, you don't need to do anything extra. I put that I'm using a licensed cord in my IND. Well, guess what? Just because you say you need a licensed cord doesn't mean I'm licensed to sell it to you for your indication. So it's very important that you understand what your licensure says and what you can and cannot do within it. And you may need to amend your BLA or refer them to somebody with a BLA that supports that intended use. The other thing I'll caution you is that saying that you need a cord from a licensed bank is very different than saying you need a licensed cord. So be sure to distinguish those two in your conversations. Lastly, I want to talk about a sales process for these types of things. It's very important that you recognize that your platform technology is really a drug, and so it cannot be treated as a commodity. You do not bundle, you do not mark up to mark down, you do not compete on pricing. You are providing something that is highly controlled, that is meeting the expectations of your client's specifications. So don't try to go out and sell. It's unethical, but also it's counterproductive, because really, if you're competing with other groups, you're not supporting the industry as a whole. Prior to making any commitments, it's really important to understand what the client actually expects from your product and what their specific requests are. There are a lot of times where people come in and they want 15 different things and I can only do 2 of them. And I say that's fine, but it's gonna cost you $500,000 and take me 18 months to set it up. And the conversation usually stops with, well, I'm gonna go find a different starting material, which is fine. You know, I price things so that we can reinvest and grow on our own, but you'll find that a lot of clients want to walk away until you've already done that investment yourself. Also, you won't find clients who come up to you and say, gosh, you know, I'm gonna need 400 cords a year, so I'm gonna help you start a new collections site. So it's very important that you're investing in yourself to grow your own cord blood bank rather than relying on customers to invest. You also need to be very transparent in what capabilities you don't have, if you're willing to grow them and what it will take to grow them. So are you gonna do tech transfer? Are you gonna buy me a Clinimax or a Prodigy? Are you gonna help me create an ISO 7 space? If the answer is no, well, maybe I'll do it on my own eventually, but we're probably not the right fit. And then don't be afraid to say no. Cord blood bankers, I tend to find, are do-gooders and not really looking at the business end of things. So it's perfectly acceptable to say no or to give a referral. I'll tell you, I hate cell culture. So I'm not going to take on any cell culture related things, but I know other banks that will, and I'm happy to make a referral, because if the industry is growing and cord blood usage is growing, it's going to create a much healthier environment for public cord blood banks. And the other thing is that it's still very important, even in these spaces, independent of where we are now, where we get a lot of bad actors who say, oh, well, you know, you can just sell me a clinical cord and I won't tell you what I'm going to do with it. It's very important that you understand what these clients are doing with your cords and that you don't sell to anyone who is maybe doing a clinical trial in another country that doesn't quite meet your expectations, that doesn't fit into your vision of how you want to grow and doesn't align with your ethics or your mission. So that's all for now, but I hope we can have some discussions about these things later. So with that, I'll say thank you, and we'll move, I'll give it back to Wouter.
I got it.
All right.
I made it. I'm okay. How does it work? Good afternoon, everybody. Donna, thank you for inviting me on the panel. And I think this has been a great meeting so far. It's been really encouraging, but we're still quite niche. We all know each other here. I can't actually see you, but I know you are there, and I talked with most of you today or in past days. What I will be talking about today in the context of this panel is leveraging cord blood operations to diversify cord blood utility. And at the Cleveland Cord Blood Center, we've decided to do that in the context of a subsidiary, which we've named Cleveland Cell Therapy Incubator. We can use some branding support. I've not been the greatest in defining very unique names for our product, which is called CleveCord or Cleveland Cell Therapy Incubator. If you have any suggestions, we're open. You know, we'll listen. So what does that mean? Let me first talk about the mother organization, the Cleveland Cord Blood Center. It was established in 2008, so we're approaching our 15th anniversary. We're the only public cord blood bank in Ohio, and we're also an independent organization, meaning that we're not under the umbrella of a university or a hospital or a blood bank, like the example that Christina presented. That has great benefits. It has great benefits for Marcy and I in terms of having freedom to make strategic decisions. But there's still bills to be paid. So we also need to be smart about how we invest and how we sustain ourselves. So within the context of the Cleveland Cord Blood Center, we got our FDA license in 2016, AABB and FACT accredited. So we have developed a track record of knowing what to do to develop compliant or regulatory accepted procedures and operations. To date, CCBC has collected over 70,000 units. There's 3 major areas where we collect: Cleveland, Atlanta, and San Francisco. Marcy has been the driving force for that. And those areas have been targeted in large extent on the patient populations there. So recently, the Bay Area operations has been opened there to get access to the Asian population there. So transplant has been the traditional utility and clinical indications. And out of our inventory, actually right now it's over 750 units have been distributed for transplant all across the world. And again, this is for leukemias, lymphomas, malignant blood disorders, and otherwise. So what you can see there on the timeline on the bottom is that since our licensure, we got our licensure in 2016, we've opened some additional sites, but we've also started opening up new departments, new labs, and subsidiaries. And I will talk a little bit more about that. So the subsidiary that I'm involved with, that I'm in charge of, the Cleveland Cell Therapy Incubator, is a subsidiary of CCBC. So what does that mean? Well, it means that it is a separate entity, but it's closely tied. There is operational contracts, and there's crosstalk. The subsidiary is managed by a board of managers, but the ultimate responsibility and governance of the subsidiary is the board of directors at CCBC. Why go through all of this? Well, it is a mechanism to take on new projects, new utilities for cord blood that the mother organization not necessarily could handle for whatever reason, or there's a benefit to do it separately. So the CCTI really, it's leveraging that expertise that CCBC has in collections, in product and process quality, collections, so there's access to our collections, the inventory, but then really with a focus on novel utility. And especially in the rapidly growing cell therapy space, cell and gene therapy space, to be able to help developers and to support new cell therapy products and developments of that. So as said, the subsidiary, the relationship with CCBCI is formalized through agreements that include operating, resource sharing, and supply agreements. And the incubator has a financial responsibility towards the mother organization as well. So the mission, closely aligned with CCBC, is advancing newly emerging cord blood-derived, cord blood-related cell therapy as standard of care for degenerative diseases, not just transplant. And those diseases with high unmet medical need, and also to support the long-term future and sustenance of the Cleveland Cord Blood Center, its mission in financial and other ways. So what is the vision to go along with that, to facilitate the production of safe and high-quality, innovative cord blood cell products ready for treatment of patients? So that encompasses the whole spectrum of being able to source through Cleveland Cord Blood Center and then to manipulate or manufacture according to needs in a GMP-compliant manner. I'm kind of hesitant to use the term GMP after the lesson we learned from Christina here, but I think you guys know what I mean. So, and the values, quality, integrity, reliability, innovation, and collaboration. For us, actually, it's that last part that is the most relevant. And we know we're in Cleveland. We know who we are, what we are. We're a small cord blood bank. And we have limitations in terms of capacity, but we are good at what we do. And we know that we have something to offer in terms of our sourcing and in terms of how we can do new things with that. So Christina also mentioned know your partners. For us, it's also about know yourself. And even though you're small, you can still be very effective and productive. And there's incredible opportunity in our field of cell therapies. So this slide here highlights several of these trends and opportunities. And what you can see there in the top, that's highlighted, I don't know how you call that shape. It is the traditional CCBC banking paradigm. So there's collections, we process according to accreditation accepted or approved procedures, FDA accepted procedures. We bank, we do banking. And then there's distributions, and distributions is with respect to transplant. The units that we collect but do not qualify for processing, we have a responsibility to send those out for research, and that is related to the HRSA funding that Christina also talked about earlier. So what have we seen, and what you all know this, what has been happening to the transplant field with respect to financial support, federal support, HRSA funding? We should be prepared to rely on that less and less. And definitely the utility rate for transplant is dropping, and it has dropped significantly because of the pandemic, but the trend is there since before that. Now, what we have seen at the same time is that the demand for research units is only growing. So there clearly is an interest, there's a demand, there is a use for cord blood and cord blood collections. So and those are based on new opportunities, new developments. So how do we harness, how do we exploit that? So we go outside of that traditional box, then you see several blue arrows, meaning those are the areas where we have seen increased demand, increased opportunity to work with us. Sometimes it's just for sourcing of resource materials, but it is also evolving into using our know-how for manufacturing, etc. And that's where we see a role for the subsidiaries. So it's to support manufacturing, GMP compliant. Is that correct? Can I say that? Yeah. And we have seen an incredible demand. I'm sorry to keep making that same joke, but we have also seen a demand in the need for research-grade isolated CD34s from cord blood, and we cannot produce enough of them. So it is an area, an opportunity, and Christine also mentioned that earlier. So CD34 is the first paradigm. There are opportunities if we play it safe, if we strategize smartly for it, where we can use the unused products. Some of them we send out for research, some of them we use internally, and we do something with it. We harvest something out of it that we can provide to the research community or to the industry. Another arm of activity where we have found demand and a request for is to provide, and I think I'm wrong here, it is fresh clinical grade cord blood units. What I mean by that is that we get requests from manufacturers, from cell therapy developers, that they don't want to rely on the frozen products. They want to get their hands on fresh units that have gone through donor eligibility, that are amenable to clinical use once they have completed their specific needs. So we have also instituted procedures to do that. What it means is that there's risk involved there, because as we all know, in order to complete your donor eligibility, to complete your product characterizations, there's a lag time in which you're waiting for your results, etc. So we can't guarantee up front that the provided units are actually eligible for clinical use at the end. This is an example of where you need to be upfront with your collaborators, and you need to be clear about what your risks are, what their risks are, and and how frequently you can provide this. This is another thing that you need to be very clear about up front. Now, there's 2 arrows there next to frozen units in inventory for alternative use. And so we have 10,000 units in our inventory. There's subsets of those that are very unlikely to ever be used for transplant unless one of the exciting expansion technologies take off and may facilitate that. There's parties interested in that because, you know, we've characterized them. We know what the quality, according to certain criteria, of those units is. The sense I get is that for certain developers, that is the current focus for sourcing. And the question always is, how many of those units do you have? Can we rely on that once we get approval and we get ready for our commercialization? And my response to that is no, unlikely. Unlikely that we have enough banked right now, or that all the cord blood banks have enough right now. And really what needs to be considered is a retrospective, so what is available in inventory, and a prospective sourcing strategy. And in the prior session, prior to this one here, I asked a question about prospective collection strategies. How do you ensure that you're collecting the units for which there is a need? I don't think there are any answers. I certainly don't have the answers at the moment, but I do think that, as we all expect and as we all hope, cord blood products will become approved, will become effective, and there will be a push to ensure source materials, whether it's fresh or frozen, and we need to be very thoughtful and smart about our strategies in commitment to collections and what units are needed, their characteristics, their backgrounds, etc. So I think it is a two-pronged approach, and that's my message to the developers that come and talk with us. Okay, so this is our incubator facility. It's small. It has its own independent clean room. It's outside of the facilities of Cleveland Cord Blood Center. It's next to it, but it's separate, so it doesn't get in the way of our licensed, of our FDA-approved process. And it also highlights the scale we can support early. We can support process development. We can support small-scale, early clinical grade product manufacturing requirements for early-stage studies. We know we are not Lonza. We know we are not the big manufacturers, but we can certainly help developers to get there. We're small organizations, and we're having these discussions with other organizations that are similar makeup as we have. So future directions for both CCBC and its subsidiaries. So optimize our inventory. So that is really, what we already have. This is what we have. How can we make sure that it is getting used for a good purpose? So identify industry drivers. I need to acknowledge Marcy here because she has been thinking about this for a long time, and some of these bullets are hers. I'd like to pretend they're all mine, but some of them are hers. So industry drivers, still the bigger is better. That is still the concept at the moment. More TNC. We know for patient-designated strategies that HLA typing is relevant, but there is also ongoing activity in universal or third-party donations in which the HLA composition is not that relevant. Reduced cost of inventory acquisition, so that goes back to the prior discussion. Streamline processing procedures, optimize collection strategies, not only in terms of logistics and staffing, but also in terms of knowing what products are attractive to our current needs, to our mission, and to future use. And again, I don't think there is a clear answer around it. I don't think that, and it is something that we do need to address, but thoughts are around, are there biomarkers or predictors that can help you assess that your product has certain predictability, certain success rate for use in this area or another. And it ties back into your product characterization. And that's where there's a need for development and for basic research. And to develop clinical pathways and product pipelines in-house. So there's also a research and development group in CCBC and support external researchers and make research cord blood units available. So we're very impressed and pleased with Christina's presentation, because we started as a public cord blood bank from a mission. It didn't start from a business-driven strategy. But we all need to grow up, and then we need to put our big boy pants on, because if we want to survive, we need to start incorporating some of those principles, right, that Christina outlined with respect to how do you define your pricing, what are the variables, what is your margin, when does that change? And we appreciate that very much, because we want to be in a position to enable and to provide in a reasonable manner the demands of the different parties. This is my last slide. So leveraging the existing resources to identify utility and to utilize our past experience. That is really it. We know how to do certain things. We have diverse partnerships. We work with academic institutions, we work with biotech, and we work with distributors for biomaterials as well. And supply chain management is part of it, but it is not the only part. And I think that if there's one message I would like you to take home from my presentation, is that I think that these new services, these new utilities, they include supply chain and then what you do with it. So there may be a marriage, a merging of supply and technologies, but there needs to also be a forward-looking component in not only how you can use what's already there, but what you need for the future. And that is something that we want to incorporate in that discussion as well. And that is part of the discussion here in this group today. So thank you for your time. I'll be happy to engage your questions later on.
Awesome. Well, I'm shorter, so I'm going to put down the microphone. I'm Heather Stefanski, and I'm excited to speak to you today about opportunities and challenges of using public cord blood units for platform technologies. So at Be the Match, and I think thinking about the missions of both Christina and Wouter had talked about, we're very aligned with our mission and find it very important about how we move forward with technology. So our mission is we save lives through cellular therapy, and really we want to create equal outcomes for all. And by doing that, we want to democratize cell therapy. And as we all know, cord blood is very important in this mission. So in thinking about cord blood, how do we get more physicians to use cord blood? How do we make sure that gene and cell therapy developers understand cord blood? It's really about education, for one thing, and also letting them know about alternative uses of cord blood that we've already talked about. And then truly, how do we have cord blood bank sustainability? So these are all very important things in order to move the field forward. So when I was a pediatric bone marrow transplant physician at the University of Minnesota, I exclusively used cord blood unless I had a matched sibling transplant, which was interesting going to the NMDP and hearing about, live unrelated donors. And the reasons that I used it is for the reasons that are listed here, but when I was treating the kids, I knew that cord blood had less of a chance of getting graft versus host disease, and it required less of a match. than typically an unrelated donor, although that is evolving as we are speaking right now. And I actually chose mismatched unrelated cord blood for some of my high-risk infant leukemia patients. So in those situations, I chose a 4 out of 6 cord because I knew that the morbidity and mortality in those patients was so high, and they needed really great graft versus leukemia effect. And that's one of the benefits of cord blood, which is what we've also heard in MRD positive patients. Whoops. This is not working. Oh, there we go. Thank you. So one of the other advantages of cord blood is that it's off the shelf. It's readily available at the NMDP Be the Match. We have cure-ready cord blood units that have already been typed. And the beauty about cord blood is that you know a lot going into it. So you know what the total nucleated cell dose is, you know what the CD34 count is, which we've seen to be more and more important, what the viability and what the volume of that bag is, the blood type, HLA type obviously is very critical. The unit age, donor eligibility safety profile, which is lots and lots of pages. I've had to go through many, many of them. And then whether the unit is licensed or unlicensed. And when I was a transplant physician, to me, you know, I used probably hundreds of unlicensed cord blood units because any cord blood unit prior to 2011 was unlicensed. And those were the units that were appropriate for my patients, and they saved many lives. So I'm grateful that we're still allowed to use those. What has also been discussed here is what are the trends in allogeneic transplant. So this was the typical, always thought of use for cord blood. And really, we've seen a decrease in cord blood use for transplant. So in unrelated, this is showing a graph from the CIBMTR with on the x-axis is years and the trends in transplant, and then on the y-axis is the number of transplants per year. And in orange is unrelated donors, whether it's, there's the thing, whether it's bone marrow or peripheral blood. This mouse is going kind of crazy, but you can see that over time this has increased. Matched related donors has actually decreased. Other relatives, which is considered haplodonors, has increased. And then we've seen a decrease in umbilical cord blood use for transplant. So what this means is what else can we use cord blood for? Well, one of them, as we've heard about here as well, is these emerging cell and gene therapy technologies. And these have increased and have the potential to increase exponentially. So again, years are shown on the x-axis, and then numbers of patients that are receiving these cell therapies are on the y-axis. And looking at all cell therapies, potentially by 2025 or 2026, 50,000 patients could have received these therapies. Whereas, you know, with the hematologic malignancies, it can be up to 20,000. And these are truly life-changing. I remember when I would have to counsel patients, my patients that had relapsed after transplant, if they had pediatric B-cell ALL, prior to Kymriah, there really weren't good options. And the risk, or the chance that that child was going to survive was probably less than 10%. However, with Kymriah, that has completely changed the landscape. It's much different now, and it's going to continue to evolve. And I can see that happening to lymphoma and so many other things that we don't even know about yet. And the exciting thing is that there's a continuous growth and investment in these emerging cell and gene therapies. So CAR-Ts are shown at the top, and the way that this graph is preclinical is in gray, phase 1 is in pink, phase 2 trials are in the light blue, phase 3 is in the dark blue, and then marketed is in green. So you can see there's not very many that are marketed, and there's not a lot in phase 3, but there's tons of these therapies that are in preclinical phases as well as phase 1, and a lot of these are with cord blood. So cord blood currently is being explored as starting material for a variety of cell types, including natural killer cell and CAR NK cell-based therapies, T-cell-based therapies, iPSC generation as a starting cell type for a variety of downstream therapies, expanding stem cell therapies, And when I was at the University of Minnesota, I actually used expanded cord blood cells to make both T progenitor cells as well as innate lymphoid cells. So T progenitors were going to be used to prevent infection as well as potentially mitigate relapse. And then innate lymphoid cells, type 2, were to try to prevent GVHD. Hopefully we'll see fruition of those to a clinical trial someday. But I know firsthand how exciting it was to be able to use, especially expanded stem cells. And T regulatory cells are also being used from cord blood. And then there's also growth in clinical trials and in investment in emerging cell and gene therapies with allogeneic therapies. So the advantages of using cord blood as a starting material is that it's available. We know what it is. It's off the shelf. You could easily grab a unit. And it's a more naive cell state with less chance of undesired mutations. And then cord blood banks obviously have lots of knowledge about the regulatory requirements of an FDA-approved product, which I hope that was the right terminology, Christina. And they're also at a lower risk of undesired mutations. So cord blood's a naive state. They haven't had any exposure to the environment or viral exposure, and they're truly more malleable because of the fewer T cells and more stem cells. And so these are an appropriate product to use. So at the National Marrow Donor Program and Be the Match, we have over 30 years of global leadership in cell therapy. We have facilitated over 119,000 transplants that we've managed, over 36,000 annual cell and blood shipments, over 6,600 annual cell therapies managed. And when I think about, I came to the NMDP last May, sort of in the middle of the pandemic, and to see what we had done during the pandemic to be able to deliver cell therapy products to patients that needed it, it was really remarkable. And then hearing today about the Ukraine and what happens in, when there is an earthquake and other things, I mean, it's just impressive about what we're able to do. Where, on 9/11, which is tomorrow, we were one of the only we were able to fly a plane that day because a patient needed a life-saving therapy. So it's pretty amazing about what we can do. We have over 22 million donors on our registry. And the other important thing that is apparent everywhere is how important research is to move this field forward. If we didn't have research, we wouldn't know that, first of all, umbilical cord blood could have been used for a transplant, or that it can be used for all of these therapies. So it's really exciting to be part of research. We have over 1,000 employees, and then our annual revenue is over $450 million. So we wanted to extend our mission, which is, saving lives through cell therapy, by providing biopharmaceutical companies with access to the cell therapy ecosystem, which has proven to successfully deliver over 119,000 transplants. And so we also have Be the Match Biotherapies. And we're focused on rapidly developing, streamlining, and delivering solutions to ensure the advancement of cell and gene therapies. So we really want to help them get to the next step by marketing and using our amazing infrastructure. So currently we have partnerships with more than 40 cell and gene therapy developers. Here is our ecosystem, and you can see it involves a lot of different things. So just from account management to sales, and then the logistics support, as well as our apheresis and collection center network. So we have partnered with many apheresis centers and collection centers and are able to say, oh, you need a slot, we can try to get you a slot. We also have our own Be the Match Seattle, which is pretty exciting. And then we can also, if biotherapy companies want to have an allogeneic donor, we can work with them on finding donors for cell therapies. So what is the value from working with us from the cell and gene therapy standpoint? Well, they have visibility into our cord blood inventory, which is supported by a lot of the cord blood banks that are here. So we have 268,000 fully characterized cord blood units from our domestic member cord blood banks. And then we also have a streamlined contracting and qualification so we can leverage our existing relationships and agreements with our suppliers. We also have the ability to offer additional characterization of units, such as gene sequencing or KIR testing or potentially CCR5 negative, which could be important for HIV patients. We also have bioinformatics support provided to help identify units. Our bioinformatics team is amazing. And then utilization of our, frankly, our logistic model. I'm impressed with them every day, as I've said, and we're able to transport cord blood units everywhere. So what I want to also talk about is the Cord Blood Bank Alliance, which we wanted to leverage our relationships with companies because they're asking, how do we get units? Can we get some cord blood units? And we wanted it to be easier on the cord blood banks because for them to have to sort of compete, as they have talked about, with different gene and cell therapy companies, that's time consuming and kind of detracts from what they need to do, whereas we can take that out of the equation for them. So the goal of this is to facilitate growth in the use of cord blood as starting material for emerging and cell gene therapies. And we want this network of cord blood banks formed to support them. So we used existing members of our NMDP network, and they have to be willing to knowledge share with our fellow alliance members and the NMDP to propel the industry forward. We want this to move forward. We want these cord blood units to be used. We don't want cord blood units sitting in the banks for 25 years. That's not beneficial to anybody. So we have this would be a new contracted relationship with us for the purpose of supplying cell therapy and gene therapy developers with units. So there's a master service agreement to govern these activities, and then riders to cover individual client opportunities. So the value to the cord blood banks is it's a single entity for contracting and quality system engagement, so they don't have to worry about that. And then we can facilitate best practices and product development to increase usage for this emerging industry. And then also Biotherapies then could provide the marketing. So they don't have to go out and market and try to get these clients. We will do that for them and distribute and have thought leadership and opportunities through our existing channels and infrastructure. And then the value to the developers is they also have one source. And I think what's really exciting, and I don't have slides on it, is that when we have this Cord Blood Bank Alliance and we know which cord bloods are part of that, a gene and cell therapy developer can come to us and say, I want this characterization of a cord blood unit. And it's not going to necessarily be coming from just one cord blood bank. Stephanie Fingerson has developed this amazing algorithm where we say, okay, you want this CD34 dose, you want this HLA type, and we search. And then these units come up. They're not identified with a cord blood bank specifically, or they won't know which cord blood bank it is. But then, so we can say, okay, these are the units that are available. And then if we went back in and did that same search, it wouldn't be the same units per se. I mean, it would depend on how many units were available. If it was a small number, it would. But let's say there were 1,000 units. It's able to really use all of these units from the cord blood bank. So I think that's really exciting. So the roles and responsibilities. So for the cord blood bank, to allow visibility into their consented cord blood products for the development and commercially available units, to permit insight into the quality systems via audits and manufacturing processes, as well as testing menus. And then to consider, what if there's a better way to do this? Things might come up, and to support these novel therapies. And then to provide independent subject matter advice and expertise regarding cord blood banking procedures and processes. Because I think, one of the frustrating things I think for everybody is that everybody maybe does it just a little bit different, and then if you want to do something a little bit different, then that can create a lot of work. So potentially standardizing this would be really beneficial. And then to participate in meetings and contribute to working group activities, and present data. Like, that's what our goal is. And then from the NMDP Be the Match Biotherapies, our goals and responsibilities are to educate and promote cord blood as a starting material through the development, engagement, and marketing teams. So to get our name out there, to let developers know that, hey, we have this Cord Blood Bank Alliance. These are the therapies that are already being used. How can we help you? We want to maintain a searchable database for that, and also, make it robust enough where, let's say, which I think we'll discuss in a little while, this is an ever-changing field, and so there are going to be things that come up that all of a sudden we actually want to look at. So then we need to be really amenable to that and malleable that we can do that with our system. And to manage the contracting and communications with the therapy developers. So hopefully then that will let the cord blood banks do what they do best and not have to be bogged down with all of these meetings. To manage the logistics, support with the necessary tools and resources and data to fulfill our outcomes, and then to provide NMDP subject expertise on this, and really to manage the invoicing. And we will charge the developers a service fee for our facilitation. I just have to put that out there. So what we won't do, so I really liked how Christina said, you need to know what you can do, but you also need to know what you can't do because you need to put up guardrails. So we're not gonna supply cord blood units for purposes outside of cell therapy development, and that does not align with our mission. That is very key. If it isn't going to democratize cell therapy, we are not going to do it. We also are not going to supply cords to clients with, if there's no intent to file an IND. Why would we do that? They can get them from somewhere else. And also proceed where biotherapies doesn't add value. So if a company needs one unit, why would we go through this entire process? They can do that directly with a cord blood bank, which would be relatively easy. We're also not going to supply fresh cord blood units. I'm going to say at this time, you never know, that could change. But right now, we're only going to be doing the frozen units, and we're not going to let it supersede existing agreements. So here's our current cord blood offerings. So we can utilize our NMDP bioinformatics, which is pretty amazing. So non-clinical use. I don't know if I should say all this stuff after Christina gave her talk. But anyway, so these are units that really cannot be infused into a human, but you could use them for research purposes, to validate processes, qualify things, investigate your internal things, and assays or logistical run-throughs. And then we also have clinical and commercial use, so we have licensed cord blood units and unlicensed cord blood units. And then our key outcomes and deliverables are to define what is a high-quality cord blood product. We want to share and consolidate the best practices really for the field. That's the goal. We want to comprehend and mitigate differences in a clinical model from a commercial setting, because that's going to be very different. And then consolidate consent, right? Consent for this is going to be different than just your typical transplant. And what do we do with that? And then consider alternatives, alternative banking, testing, and attribute models to support these novel therapies. And then, as I said, we really want to disseminate this information and show, what did the Cord Blood Bank Alliance do? Like, what does this mean? Did we get out best practices for shipping? Did we get out best practices for whatever it is? And then how many gene and cell therapy developers did we give to? And then also, I think what would be interesting is to think about, what, from a gene and cell therapy developer perspective, what did they ask for? What were we able to get them? And what do we need to do as a Cord Blood Bank Alliance to continue to propel forward and maybe change some of the processes? So here are our current Cord Blood Bank Alliance partners: Bloodworks, Vitalant, Cleveland Cord Blood Center, South Texas Blood and Tissue, Cord for Life, Versity, the Carolinas Cord Blood Bank, and STEM Site. So thank you very much for your time. And I'm excited for the discussion. So, Christina, sorry. So I loved a couple of your slides. I loved how you told us exactly how you price cord blood units. And so I'm gonna ask these questions first. Do you see a lot of variability in your prices? From like, first of all, how often do you look at that at your formula? And then in the last few years, have you seen big variabilities in your prices of cord blood units?
Yes, so I should say beyond that, I stratify my cord blood units. So I don't have one type of research unit, I have 4. I don't have one type of processed unit, I have 3. And that's an additional to units that are banked for transplant. And I'd say due to variations in the number of cords collected and the number available for each type, I've probably seen a 20% plus or minus over the last 12 months looking at the year over year. So I don't necessarily reprice things, but I keep an eye on things, and when the margins are too low or when we're taking on a new project that needs investment, I will reevaluate the pricing.
Awesome. And then my other thought, or the other interesting thing that I liked was when you showed the graph that showed the different cord blood banks and then what they did. And so in looking at that, what do you think the next things are going to be that you're going to be looking at on cord blood units? Do you have any insight to that? Or even Wouter, like if, you know, what you're looking at now, what are you going to be looking at? You know, are you going to do some additional typing? Like, is KIR going to become standard? Is CCR5 going to become standard? Or, you know, in other things?
We have continuously looked at that, and I've been looking at that for the past few years, and that was independent of you know, a direct demand or a need. But we have implemented CCR5 testing years ago because it was there, and it just made sense to do it. We are interested in the type of potency assay development that our friends at HEMA Quebec are developing. That is something we would like to incorporate, because we also believe that the more you know upfront about the characteristics of your product, the better positioned you are to find the correct utility for it. So that is really focused on utility, not necessarily on pricing. Coming back to the pricing, the system we've used at CCBC is that there's a certain expectancy around for different cohorts. So you have smaller units, you have bigger units, intermediate ones. We know what the frequencies are for selection of transplant. We know what the revenue from that would have been. So we use that as a reference point to identify how much we need to recoup from individual units that people are interested in. So in that sense, there's a sliding scale. We haven't changed our pricing necessarily for individual cohorts, but not all are the same. But what we would like to do is learn from Christina and align that with, you know, periodic assessment. And I do think, even though we're all different, I do think that it is relevant to know where those pricing levels lie across the field, so that we don't undersell ourselves, and also we don't compete out our colleagues and our other cord blood banks.
And I'd have to say that my focus probably over the next 2 years will be on what information I can generate from existing samples that I have on cords. You know, what do I have for genotyping? And one of the biggest challenges outside of cord blood in donor selection is that you get people asking for things that require functional assays, and they're looking at the wrong correlates. And those are things that take years of donor recruitment and screening and multiple samples being sent. And so I think the field needs to be focused on guiding selection parameters that are capable of being taken from units that are already banked. Can we do genotyping, or is there a way we can do cell frequency on something we already have? And so I don't know what it will be beyond the things that were on there, but we need to focus on making sure that people are asking questions that we can actually answer with our existing inventory.
Well, I invite the audience to ask some questions. Hi, A.B.
Hi. So, might be beating a dead horse here, but I wanted to offer some comments and caution to some of the nomenclature being thrown around, and back up a little bit of what Christina was saying. This aspect of GMP grade, clinical grade, whatever we want to call some of these things, parallels some of the discussions that we've had in the cell and gene therapy field, probably going back to the late 2000s into the early 2010s. And you can find a lot of discussion around that in some of the articles written about qualification of raw materials, ancillary raw materials, how do you qualify if it's an excipient, et cetera, et cetera. So where I'm going with some of this is when we call something a grade, the question is, what does that mean? And so if you think of something like a USP grade, EP grade, JP grade, those are pharmacopoeia standards. You can pull out something, you can say, here's a list, and I can compare something to that agreed to list, and now that becomes a grade to a certain degree. Then you have standards, and obviously with the standards, we can look at something such as whether it's the GMPs. So we say GMP. What does that mean? Is that GMP drug 210, 211? Is that GMP medical device 820? Is that something else? So if you put something on that says GMP grade, and then someone comes in and audits you, and they go, great, I'm going to audit you to GMPs. Who's deciding what those GMPs are? But you've already set yourself up for potential non-compliance or, let's say, a disconnect in expectations to a certain degree. Not even counting what that might mean for potential liability if you claim to something and then someone goes and uses it, and they're going to turn around at some point and say, but you told me it was XX, Y, whatever. Anyway, so I would offer that as caution, and that's coming from personal experience. For those that don't know me, I'm A.B. Matthew. I'm with BioLife Solutions. So, many folks might know us from our CryoStore cryopreservation media, and how these are not regulated raw, reagents, and just replace the word reagent with not necessarily regulated raw materials that we're using and someone wants to use for that process. So really what it comes down to, it's a nuance for some of these, and sometimes folks might say, well, we're saying the same thing, but we're really not. GMP grade is not the same as manufactured per GMP. You can manufacture per GMP, and then you can basically come to some agreement, what are we saying? Good manufacturing practices, not necessarily GMP 210, 211 or 820, but good manufacturing practices, and what are we taking from that and that we are agreeing upon. So those are, I think, I would suggest the pathways for someone to look into. And then when we say something like clinical grade, again, what does that mean? It's what people want it to mean, and the joke around it is, we probably semi-created the term, not me personally. I always will say, and we're probably being recorded, but yes, it was our CEO. So it's one of those things where it comes in to make it easy when someone wants to sell something, and it brings comfort to the other side. But when that quality and regulatory person comes in, what exactly are they measuring you to when you say that? And what liability are you taking? Because what you actually did is you processed it in alignment with what you would have done for your clinical product, which means that it's manufactured and processed to a very high degree, to a set of specifications, absolutely good thing. But if you put a label on it, you're kind of saying something about it, which means that your customer, and again, these potential bad actors, these customers are going to turn around, and when they have to talk to a regulatory body, or when they inject something into somebody's eye and suddenly they're blind, they're going to turn around and say, but we used clinical grade product from so-and-so. And so it doesn't mean that you can't use some aspect of this terminology. It's a nuance. So again, a nuance. So I'm being very careful today. I'm not saying that anybody is wrong. There's a nuance to it. You just have to be prepared for what could be the potential outputs that come. So for me, part of what I would say and what we say is manufactured per GMP, but not GMP grade. And then if it was a clinical aspect, we're not directly clinical, but it's where something is manufactured to a clinical, expectation. I don't want to say standard, but it is to standard to a certain degree because you've established a standard within your own manufacturing system. So there's a clinical aspect of what can be expected, measurable, and that it's very good, but just obviously being careful what necessarily that, might mean.
So can we comment on your comment?
Yeah, just one more thing. I just wanted to say, just kind of like the extension of all that is kind of where we talk to the fact that this is all evolving. And that's one of the things that Heather was saying and that I think we're all experiencing is we're shifting from these regulations around blood and blood products to cell gene therapies, regenerative medicine, which has allowed for more flexibility. And my recommendation is that you embrace where you have that flexibility before jumping into creating a box around standards. And we want to obviously, I think, decrease the variability in the processes, but standard comes with sometimes handcuffs and expectations and everything. And so when something's evolving, if you put yourself in a box, that may not always be a good thing. It might be setting up for almost like a I don't want to say compliance necessarily, but a non-compliance, whereas best practices, which was another word that was used, sometimes is something that might establish that this isn't the wild, wild west, but that it does provide some level of best practices and good practices and things like that come in. So that's the only thing, and I'm just transferring a lot of conversation that we've had a lot of times in ISCT and the raw materials. So again, I wasn't saying that anybody was necessarily wrong, but I'm just saying you just might want to caution. That's all that I'm saying for
Yes,
raw material.
and the thing that I constantly keep in mind is that these were processed as clinical units for transplant, not for the indication that I'm talking to a person
Correct.
about. So I don't necessarily have the release criteria. I don't know the longevity of the cells in my cryopreserved unit that they're looking to use. And so I can't say in good faith that it follows those things because they haven't been validated. So for transplant, I'm happy to say clinical or GMP, but for other indications, I just have no clue yet.
But would you call it a grade?
So the other aspect is with committing to distribution and intended use. So none of this is just haphazardly distributed. There are negotiations, there are contracts, there's quality reviews underneath
Absolutely.
that. So even though we may be loose in our use of those terms here, for the intended use, it is clearly defined in contractual agreements. And clinical means that if certain criteria are fulfilled, it may be used in a clinical trial. It may be used in humans. Non-clinical or research, frequently research is equated to non-clinical, means that under no circumstance may the recipient use that material that is provided in a human being. So I appreciate your feedback, but there are safety checks. There are checks in the process that we as cord blood banks use to, enable that distribution process.
Yeah, and I don't disagree with that. And that was one of the other things that I was thinking about when it was presented was there's a lot of it that's tied to perhaps a service contract or the way that you're kind of putting it together with a client contract, which is a little different perhaps maybe for the long term, where you might want to consider if it's a product that is being brought out and not necessarily a service and then a product. And so, because especially early on, there's a lot of bandwidth, there's a lot of costs, and then a lot of one-on-one time with that person, that other group that might go in when you're doing that for the service relationship. So I was actually looking at what you're doing, and I think where a lot of folks are thinking about is if we have these units, these capabilities, especially unused units, what else can it be used for so that we can gain revenue, sustainability, things like that. There's a pathway towards that where it then becomes almost an off-the-shelf product that has already been processed, and now we want to be able to move it towards being able to sell it in revenue, and it's been characterized. That's the key part, that it's been very highly characterized. And so then when it becomes like a product, that aspect you may want and you may have it today. It doesn't necessarily always mean that you will have that complete control over when that person buys it, what they use it for, et cetera, et cetera. And it might be the difference between what you have now in a service agreement and what might need to go into terms and conditions, TNCs that might go with the purchase of something, which we do, for example, where the TNCs can indicate that if you go and use it for whatever you use it for, that's not a liability on us as the raw material supplier. And that's where it could live in. And again, it's the worst case scenario, not the best case scenario, for some of it. So where I'm going with it, again, that's why I was trying to be very careful not to do it as a criticism of something being wrong. It was more of what I think we've seen and experienced in the cell gene therapy world with a lot of other raw materials, and then where does this overlap with that aspect of raw materials and characterization of the raw materials, because this is relatively new for cord blood and cord tissue, but it's something that we've struggled with and been working through on a lot of other fronts for other raw materials and things. And it's just if one wants to, take it, so be it. If not, we certainly understand, because it's still going to be different for this.
It's definitely evolving.
Yeah,
Yes.
and that's the thing is, just so folks don't necessarily have to experience some of the pains that some of us otherwise have had to experience, especially during audits and things like that. And that's really where it's more of a feedback for consideration.
Thank you for the tips, Aby. Nairi?
So thanks for a really interesting session. My question's for Heather, and just wondered if perhaps other international cord blood banks were wondering. So the AusCord banks have an IND with NMDP for export of cord blood into the US. And I suppose just thinking about your cord blood bank alliance, is there opportunity for international cord blood banks? Yeah.
So currently we're only doing domestic, but I don't ever want to say that never is never. So as we're getting it together and figuring it out, I would say international will come next.
Okay.
Yes.
But thank you.
Yeah.
We want to see how it works first.
Yeah.
We got it, yeah. Let's make
Marcy?
sure it works.
Yeah.
So thank you all for such great talks. I think it's really important as the banks think about, the new opportunities. And I think the Cord Blood Alliance is a great opportunity for us to really think about new uses for it. And one thing that I've been involved in the Cord Blood Alliance a little bit in the onset, but Heather, as you were talking, I was thinking that one benefit of it that I hadn't thought of before was that the Cord Blood Bank Alliance will have an opportunity to see what that need might be like going forward. Because
Right.
one of the big challenges for the banks, Wouter brought it up in the last session, is us trying to plan. So
Right.
we have collection sites. We're trying to be really thoughtful about utilizing our resources. And, we map our developers we work with and where they are on the pipeline. But I think if we can provide that information on a larger scale, because you'll probably see more developers ideally than we would as one cord blood bank, I think that information would be really helpful for us as banks as we're trying to plan our future supplies. So I think that would be really
I totally agree. And I think, as we go forward and say, oh, all of a sudden, they want us to isolate NK cells, so then potentially other cord blood banks then might want to start doing that, or it could be many things. But yes, I totally agree. It's going to be really exciting.
Right. So sorry we didn't have more time for questions, but I am hearing a couple of themes. One of those is standardization. In the previous talk, we had consolidation, perhaps, to maybe, create some scalability that decreases the costs and provides efficiency. So I think that's really important. Roberto has talked about simplicity, bringing growth to us, and I think that's a great thing. And Filippo, we heard him first day talk about what's your niche, and so I was really pleased to see, Christina, that you had included that too. You can't do everything and do it well, so figure out what you do well and go with it. So we look forward to the future. I think this is a really positive advancement, and we will see fruits of our labors here very soon, I'm sure. So thank you for your attendance today. We'll adjourn.